Welcome to the Knowing01 Multiomics webinar series.
Welcome and thank you for joining us today in our Multiomics webinar series. Today, our CEO and founder, Nicola Miller, will talk about the complex journey from data generation to patient outcome and how to connect all the dots before doing investments. Please start, Nicola, the stage is yours.
Thanks, Caro, for the nice introduction. As always, welcome to our Multiomics webinar series. Today, we are going to talk about anything from model to patient, basically how we are able to connect the dots before we actually commit to a clinical strategy. So we all know about the outcome of drug discovery is not in its best shape, right? We have a lot of hidden pitfalls along the path, and with looking at some of the real world examples here, you can see even the best names have failed programs. It is not about hitting primary endpoints, but also here you see with gene therapy, you also have some death involved. So there's quite a lot of patient biological risk that comes with the business risk of actually developing new drugs. If you're going to look at the process of this drug development, so where we are really starting the journey to ending up somehow, somewhere at the patient benefit, it is many, many steps involved, and I'm just really trying to highlight only a very few here. But let's say very high level summarized, you have a safety and toxicity track, and that really has a lot of company and risk involved, where we have really the safety, the preclinical candidate selection, which can be, for example, small molecule or an antibody. It's also about making sure manufacturing could be possible, and you also then have to bundle this to an IND-enabling package to submit and start phase 1. Also, many start with a disease biology, so the disease impact assessment. Some do all or only parts of it, but nevertheless, it's about really trying to understand the disease, because the target or the drug or the therapy that you're developing should modify the disease. Just to embrace that, it needs to be disease altering, right? And if we're doing mistakes there right at the very beginning, we will not ever be able to have a patient benefit. Now, of course, for phase 1, you also need justification of the clinical strategy that involves having an idea of the first indication, or justification where you start with your indication, search and treatment. Disease model selections are part of it, but also early, or let's say an initial idea of which patient to treat, clinical feasibility, dosing, of course. So again, more steps are involved, but just to have a nice summary here. So once we then go through phase 1, and hit phase 2, we also need additional packages and information. So we actually need a clinical translation strategy. We need to make sure here that also with the right dose, with the right therapeutic, that it's possible to treat a patient with a specific indication. Also, biomarker discovery is quite an important step here as well. This all feeds into phase 3 clinical trials, where we really have then also the topics of enriching for population. So we really have to go to patient plan. That especially also comes back with CMC manufacturing, GMP. You really need to make sure that it's actually doable to treat a patient at scale. And then once assessed, we hope we have a nice or good outcome at the end of the clinical trial. So you see a lot of steps, but there are many steps in the beginning that are really pivotal and are only turning out to be successful or non-successful after 10 years after the clinical phase 3, for example. So where we come into play, and that's why we're also having this webinar today, is that at many steps along the way, a lot of decisions you have to make. Not always you have the clarity that you need. So our claim here, and we've been doing that with every partner we're working with in biotech space, we are trying to add clarity where others stall. What I mean by that, it's about avoiding running after the false signal, or just running behind a noise where you think it could be a nice modifying drug, but at the end of the day, it doesn't really do what it should be doing. It's also about trying to improve phase 3 success odds, and also bottom line, because it's a lot of venture created business here, it's about building a stronger partner ready story. So why do we talk about this at all? I recently heard a biotech CEO, she said it's basically because we cannot afford to guess, we cannot make mistakes along the way. So let's try to make use of what we already have, right? To complement to the biology at hand. So this is where we also come into play with knowing01. We're trying to really support our biotechs and partners with the technology excellence. So what do I mean by that? It's about taking a decision on substantial amount of data. So did some primary market research, and around about 5 to 20% of the actually available molecular data is being used today. So what we are saying here is that nice slogan of never missing the forest for the trees, about the context as the key to success to really find the real signal, the real deal that really alters the disease biology and it actually matches out. At the same time, it's also operational speed, because every month you are earlier on the market is a lot of more money and quicker return for every of the investors. What we basically see that 6 months or more companies tend to take for decisions where a lot of unstructured data is involved. Some even don't get started in assessing more data and validate. So we can really be, with our automation platform, be super quick and really cut several months even from the timelines for decision making. Well, we do this by linking data, models, and also analytics across the entire development process. The slogan here that we're using is basically be smart, reuse, improve. There's so much data out there being generated, put into the public domain, being within the company already sitting fully unexplored. So with the right technology, you can be really super smart, but also with a nice substantial amount of data, take the decision in an easy way. I want to give you basically one example where we can do this quite nicely is on the selecting an indication for the early clinical strategy that ideally best matches the mode of action, so the MOA, to increase patient outcome. We often start working with companies where it's about making sure either backing up the first hinge of an indication that might work or screening for different oncology indications, for example, or autoimmune indications, trying to see which one is actually the one that works best when I modify a specific target or a pathway or whatever treatment modality you might want to have. So basically, we are going to contextualize step by step the indications and imagine every of these indications we find a similar amount of data space. So we really can fish for evidence or find evidence, and then we can prioritize and score, say, okay, which is the target and pathway match, so my mode of action of an indication. And here, for example, you see two differences visualized very simply. Basically, an indication A has a high priority because you see a lot of signal, a lot of regulation that is very key to the disease, while comparing to indication B, where we have the same context size, the evidence is really marginal at best. So you have definitely a high priority for indication A and a good likelihood that this indication might actually work best for your new drug. So I could be calling this target indication fit, where it's about in silico validation of the target and the activity, here, for example, in downstream pathway regulation. It's about comparing and scoring the interesting indications to then select an indication to put into the clinical strategy. The way how we make it work, so our magic sauce, we can actually scale with data and also in time, is 3-way. So I always like to call this here is that we are really going beyond bioinformatics. So we really try to think it through a bit differently, not about digging deep in one data set and losing yourself in p-values, but it's about systematically adding layer and layer of evidence and evidence to make sure you did your homework and you also did this roughly when going forward with your clinical strategy. So the way how we do it and make it work is about 3 things. It's about a context at scale. So we have worked on trying to identify smart data adapters, so we can really work with any type of complex data of any of the omics layers. That is part 2. We have a MultiomicsAI, which is a knowledge graph technology in our software, where we really can link evidences across the entire multiomics domain and also across model organisms or human beings. So solving this entire homology also at the same way. The third part, because we want to have a no-code solution, so those people who really understand the biology can actually read the data. We're building basically a software for scientists that are not just for the data teams. Okay, so you can really assess and understand the data at hand. We see that we can come from 4 to 6 weeks from a question to a decision. We see that we have really a 5 to 10-fold increase of the data scope. So the decision is really being made on rough data numbers. And we saw already that we can effectively de-risk 10 or even more clinical trial programs from oncology to immunology, all the way back and forth. And really it works always with the same technology, same way. And we can really succeed here and support biotechs in their development path. And with this, I'd like to thank you for your attention. Thanks for tuning in.